the opportunity to give this talk today. My name is Magda Kordon and I'm the CEO and co-founder of InterDNA, and today I would like to talk about unveiling DNA breaks through direct stride assays for precision in preclinical and clinical development of DDR inhibitors. So to begin, I would like to first maybe highlight what are the general expectations regarding the PD biomarkers that we use in DDR inhibitors development and actually beyond. So we want all the methods to be as proximal as possible to immediately report on the target and modulations. We are looking for robust methods that are going to be specific, sensitive, and reproducible. We want them to provide us with objective quantification of the signals. And finally, we would like them to accompany each stage of drug development. So when we think of DDR inhibitors development, we're targeting specifically chosen proteins from specific different repair pathways, and we're actually targeting them in cancer cells that are characterized with suboptimal capability to repair DNA damage. So the ultimate goal of this, therapeutic strategy is to actually kill cancer cells by increasing the level of DNA damage to a level which exceeds the capacity of DNA repair mechanisms. Therefore, we should consider measuring levels of DNA breaks as a unifying and proximal to PD biomarker across multiple different DDR inhibitors. And these days, we're actually using a broad spectrum of methods that are based on detection of DDR-related proteins using IF or IHC-based microscopy methods to actually assess the level of DNA damage. One of the great examples is gammaH2AX, which is considered to be a gold standard, and it's considered to be equal to the presence of double-strand DNA breaks. However, we shouldn't take DNA breaks for granted, and I would here like to maybe list out a few limitations of such approaches. First thing to consider is that the protein foci, including gammaH2AX foci, they do occur in the absence of physical DNA breaks. There is a growing body of evidence that phosphorylation of the histone may actually be a type of a response to general cellular stress, or just, for example, a distortion of DNA structure. The technical shortcomings of IF-based or IHC-based approaches, such as non-specific antibody binding, can lead to increased background levels, which can then make actually the detection of true signals more difficult. That limits the level of specificity and sensitivity of the assays. And obviously the challenging aspect of objective quantification of DNA damage level should be also taken into consideration, given the fact that the stainings may result in different shapes of foci, different types of shapes that we're actually detecting in the images, like pannuclear stainings that are hard to interpret as well. Counterintuitively, when we develop DDR inhibitors, we're using the markers that are involved in DDR, so they're dependent on active DNA damage response, whereas we're interfering with the DNA damage response with the molecules, plus we're looking at the cancer cells that are very often characterized by DDR deficiencies. So it's again making it arguable whether we should consider such signals a good reference. And finally, there is no differentiation if you look at particular proteins between different types of DNA breaks, and there are certain challenges in moving from preclinical cell-based assays into preclinical in vivo or clinical studies that have been reported. So at InterDNA, we're offering, and we would like to present the STRIDE platform. So STRIDE stands for Sensitive Recognition of Individual DNA Ends, and this is basically what the technology does. It's a fluorescence-based platform technology for direct detection and labeling of open free DNA ends that occur at the sites of either DNA breaks or, for example, different types of DNA fragments. So STRIDE is direct, it's specific, sensitive, it's compatible, quantitative. So by the fact that it's direct, we mean that we're capable of detecting and labeling the DNA ends directly in situ inside the nuclei in any type of biological material. We don't need to isolate DNA from the nuclei. It is specific and quantitative. The signals occur in the form of very bright fluorescent foci with a background close to zero. And those foci selectively label either single or double or protein-associated DNA rates. And actually having this good signal-to-noise ratio makes it very easy for us to accurately count the signals by our in-house built AI-based algorithms. It is sensitive, so it's a reliable measure of DNA damage detection at an individual lesion level. And finally, it's compatible, so it can be applied in any type of biological material that is obtained at any stage of drug development pipeline. So we've developed multiple different solutions based on this platform. One of the first ones is dStride. It's a specific assay for detection of double-strand DNA breaks. And here I'd like to just present very briefly what are the principles of STRIDE, and well, obviously it's a method, so it's a sequence of steps that need to be taken to obtain the final result. The first step is the enzymatic modification of DNA ends that then can be followed by antibody-based detection of incorporated modified nucleotides, and then finally the third stage is the signal production amplification based on rolling circle amplification reaction. Another solution like this that's based on similar principles but designed in a slightly different manner is sStride, and sStride is a specific assay for selective detection of single-stranded nicks or single-stranded gaps. But we also think of STRIDE as a platform for customized assay development. So based on the STRIDE principles, we've developed multiple different functional assays that are actually measuring the activity of specific pathways. So we're looking at coincidence of certain type of machinery at the sites of DNA damage of different types. And here are just two examples, sStride MMR and dStride HR assays that are measuring the activity of NMR pathway or HR machinery at the sites of double-strand DNA breaks. And I actually strongly encourage you to visit our poster later today, where Magda is going to be presenting the whole use case of using sStride as a platform to develop some customized functional assays, including sStride MMR. So I would like to now show you a few different examples of applications. Here are a few applications of preclinical, let's say, applications of dStride or sStride in cultured cells. And here you're looking at the detection of double-strand DNA breaks in colorectal carcinoma cell line, untreated and treated with etoposide. If you look at the red channel, there is quite nicely visible response to the treatment. We compared dStride here head-to-head to gammaH2AX. You can see that the signals appearing in the form of very bright foci. It makes it very easy to quantify the signals, like literally just count the foci in 3D by the algorithms. Another example of sStride application, here we're looking at DLD1 wild-type cells where we detected single-strand DNA breaks and actually looked at localization of RPE molecule to do the spatial correlation analysis and localization analysis between the two readouts. So we've investigated multiple different DDR inhibitors, but I would here like to maybe mention a few of the studies that have already been published. Here's the example of a paper published in 2023 presenting the application of dStride in assessment of MOA and PD effects of new generation acetylfulvene. Another example of the application of sStride and MOA assessment and target engagement assessment in the process of USP1 inhibitor development. That paper here listed is the research paper showing another example of application of dStride, in this case to label breaks in chromosome bridges and quantify them. And finally, the paper that is actually showing a completely different approach to the application of the assays, where dStride was used to look at the DNA fragments. We measured levels of cytosolic DNA fragmentation in the process, in the context, let's say, of POP7 inhibitor development. But going beyond the cells, STRIDE assays can be also applied in FFP biological material, and here are some examples of the labeling of endogenous double-strand DNA breaks in clinical patient-derived samples. These images were obtained throughout the collaboration with AstraZeneca, and here we're looking at the detection of double-strand DNA breaks in colon cancer biopsy. Another type of example where we looked at the breast tumor biopsies from different patients, and we actually quantified the signals in order to look at the intra and interpatient variability. So we looked at different areas of the tumors to look at the level of variability of DSBs and SSBs in parallel, and it's all doable and fairly easy to be performed using our algorithms. So as another, let's say, follow-up to showcasing the usage of STRIDE assays, I would like to maybe also look at some direct comparison between STRIDE assays and gammaH2AX approaches. So I would like to bring to the table a few examples of very specific applications. That's an example of an experiment where we compared gammaH2AX with dStride head-to-head. in RKO DTAC cells that were treated with DTAC13 to lead to the degradation of a factor involved in double-strand DNA breaks repair. And interestingly, if you look at the data, gammaH2AX was characterized by a very high level of variability and, let's say, low level of reproducibility between the replicates, what was actually limited and much improved when looked at the D-stride assay. And then when we put all the data together, D-stride, given its high level of specificity and sensitivity, showed a very clear response to the treatment and very clear, effect of the target engagement. Whereas with gammaH2AX, it was a little bit, less clear, and there was quite a limited dynamic range. Another type of application where we looked at FFP PDX models treated with a DDR inhibitor. We measured here gammaH2AX and saw no response, but when we specifically looked at single-strand DNA breaks, apparently it appeared to be the leading mechanism of action and actually the leading effect of the target engagement in those samples, which could not be revealed with a gammaH2AX based approach. And finally, the application of gammaH2AX and D-stride in FFP human PBMCs. It was done in collaboration with AZ. And here the biggest challenge of gammaH2AX was a very high background level of phosphorylation in untreated samples, which led actually to it was hard to conclude anything from the data, whereas D-stride showed a very nice response to the treatment, and we looked at the combination of the treatment with a DDR inhibitor with bleomycin, and with the DDR inhibitor present, we saw this nice sensitization effect and a delay in response in DNA damage repair. So as a final note, I want to mention also D-stride HR assay as an example of those functional assays that we have under development. And D-stride HR was purely designed to study homologous recombination repair in tumor biopsies. And I would like to reveal first how this technology works. It's based on the same principles with a difference at the stage 2, where we're looking at coincidence of RAD51 or RP molecules with double-strand DNA breaks. Here I'd like to maybe mention our collaboration with MSKCC and just show some sneak peek of that ongoing work where we look at 2 assays, D-stride RPA, D-stride RAD51, to measure endogenous levels of the activity of those 2 factors at the sites of DSBs in the tumors pre-characterized with different BRCA1 and BRCA2 status, and we can already see, it's not maybe very clear here in the picture, but there is a differentiation between tumors that are BRCA1 or BRCA2 mutant versus the BRCA1/2 wild type, both with D-stride RPA and RAD51 assay. And the data can be easily quantified. There is nice reproducibility. It's just a limited number of patients right now that have been characterized, but it's an ongoing work. But there's definitely a differentiation between the wild type models, and we see it as a very promising data set to further develop it as a potential predictive biomarker, functional predictive biomarker. So here's a slide with a short summary. I don't know whether we have still time to go through it. I think that the key take-home messages are just to consider the direct detection as the key driver for the PD analysis in the DDR inhibitors context. The STRIDE platform can be a useful tool for that. We are also capable of customizing STRIDE-based solutions to measure pathway protein-specific DNA strand breaks, and D-stride HR assays are actually promising reporters of HR status in patient samples. Here I'd like to thank all our collaborators, but first and foremost the DNA team. And yeah, thank you for your attention, and I'm happy to take any questions if there's some time left, and if not, then please reach out directly. Yeah.